Aloha, thank you guys for joining us for our pre-recorded Zoom presentation for Thursday, May 7th. I'm happy to have Patrick Nichols from the Zoology graduate program, University of Hawaii at Manoa, and he'll be talking to you about rapid assessment of coral reefs using environmental DNA, all this from a bottle of seawater. So without further ado, Patrick.
To be here today. So as Gavin said, I'd like to talk a little bit about my PhD dissertation work and a little master's work as well. Again, that's titled Rapid Assessment of Coral Reefs Using Environmental DNA. I'm going to talk about everything. All of this information was collected simply from a bottle of seawater. So all of this work really boils down to an advanced degree in moving various volumes of liquid from one container to another. So here's me moving water from medium-sized containers to small tubes. I'm very good at pipetting and sitting at the lab bench, moving these volumes of liquid from one container to another. And ever since I was a child, I've been fascinated with water. So here's a picture of me that my Nana took of me just staring at the water. So today I'm going to tell you a little bit about some particularly interesting aspects of looking very closely at water. So first, some background to set the stage for my thesis work. So coral reefs are in decline. Across the Pacific, reefs are being lost at a rate on average of 2% per year. And the Hawaiian Islands are far further down the trajectory of decline. So therefore, we need to rapidly and effectively monitor coral reefs. And these assessments typically rely on visual-based surveys. So you can see in the background picture, here's two divers surveying which corals are on this reef. But these techniques can be time-consuming and actually extremely costly in order to have teams of divers in the water constantly surveying coral reefs around the globe. So I began looking into using what's called environmental DNA, or genetic material that's obtained from the environment, as a possible alternative into surveying coral reefs. So the benefits of eDNA is that it can be rapidly collected, as you can see here, by just a bottle of water. We can glean all this information from the environment. It's also generally more cost-effective and less intrusive than many other survey techniques. So whereas I would visually survey a reef or pull fragments of coral from the reef, eDNA is a way that we can get genetic material without even touching the coral organism. So this genetic material from eDNA can come from a variety of different animal sources, such as sloughed off tissue cells or gametes, anything like that. Or in the case of this parrotfish, it could be this mucus barrier around it. But when I think of parrotfish, more often than not, I think of fish poop. So yeah, fish poop can also be a form of environmental DNA. And researchers have used environmental DNA in a variety of different applications, tracking everything from the spread of invasive species, such as the movement of Asian carp into the Great Lakes as part of an ecological monitoring program, which was established in 2013 in this study by Christopher Jerd and colleagues. And eDNA surveys have also been used to assess bird diversity using bird poop runoff in a study by Ushio and al. Colleagues, 2018. So this study, they were looking at eDNA from bird poop and tracking endangered bird populations over time. So a unique application of eDNA there. And other researchers have even surveyed entire ecosystems, such as this well-known study by Jesse Port and colleagues off the coast of California, where they're looking at kelp forest vertebrate diversity. So specifically what vertebrates are living in these kelp forests. And eDNA is quickly becoming a hot topic in molecular ecology, and it's even being used to catalog biodiversity in Loch Ness with hopes of exposing the long-lost monster. So the basic theory behind eDNA surveying is that small volumes of water can contain enough DNA to detect entire communities of species, and even rare species. So DNA is constantly expelled by organisms and degrades at a quick rate in many environments. So DNA from the environment can provide a snapshot of biodiversity at any given time in an ecosystem. However, there's several considerations that must be taken into account for surveys using environmental DNA. And one of those considerations would definitely be primers. So primers are short sequences of DNA that serve as a starting point for targeted detection of an organism or group of organisms. So you can see here the forward primers being pointed to with this blue arrow and the reverse section of the primers off screen. But these primers used for eDNA detection aim to minimize primer binding site differences among taxa. So we want as few differences in the genetics in this red box here among our organisms. Having too many primer mismatches in this region can actually affect its efficiency later on. So this is one of the considerations we'd like to look at when we make primers for eDNA. The other thing is that we want to maximize what's called polymorphic site differences. So all these differences in the red box here, this larger section. So the more differences that we have in this region actually allows us for better differentiation among the organisms. So we have more power in determining which species of corals, for instance, here. And these polymorphic sites are essentially what make each organism unique. So all these different base pairs highlighted in this red box are what make these coral organisms unique. So selecting or designing these specific primers using the right key is actually vital for environmental DNA surveys. And there's lists of primers that have been previously published, as shown here in this table from a paper by Michael Statt and colleagues. So primers have been designed to target DNA from a variety of different groups of organisms. As you can see here, everything from eukaryotes to metazoans, fish, mammals, crustaceans, cephalopods, plants. And in molecular biology, we design primers to target specific areas of the DNA, as you can see in this column here, with plenty of different markers representing different genetic loci for these different groups. So the difficulty in using eDNA essentially boils down to which primer you choose. We must choose the right tool for the job. And meta-barcoding is one such tool. So meta-barcoding uses universal primers to retrieve amplified DNA from many organisms all at once, such as this paper also by Michael Statt, where they demonstrate the ability to survey biodiversity across the entire tree of life. So this one-size-fits-all model may work well for the presence and absence type data, so looking at the diversity of species, but it actually makes estimates of abundance extremely difficult because these primers tend to have biased amplification of certain groups. So for instance, it might amplify rays more than it does crabs and things like that. So not all organisms match these universal primers to the same degree. On the other hand, we have what's called species-specific primers. So this is typically looking at the presence or absence of one species of interest. However, surveying entire communities of organisms using these species-specific primers can actually become extremely overwhelming very quickly. So imagine you have a primer for these anemones and urchins and algae on the reef, and then all these inverts and fish. And very quickly, you can see how having one primer for each of these species is just going to become a tangled mess and actually ends up being less cost effective than the previous meta-barcoding approach. So as a coral reef ecologist, I was particularly interested in asking, can we use eDNA to actually quantify coral abundance in a cost-effective and replicated meta-barcoding approach? So corals are ideal subjects for this eDNA surveying method, in part because their mitochondrial genomes are highly conserved across taxa, so we can actually distinguish these species relatively easy with a couple primers. And also their surface area is a good approximation of their biomass. So they have basically a 1-to-1 ratio of surface area to biomass with this thin layer of live tissue which should make comparisons of live coral cover to DNA content more accurate. Other organisms, as we mentioned, like parrotfish, which may poop a lot or shed a lot of eDNA, may not be as good for getting abundance from eDNA as corals. So secondarily, with coral bleaching events becoming more and more common due to increased sea surface temperature, as you can see here in this picture of bleaching corals in Kaneohe Bay, I was interested to see whether or not we could also use eDNA surveys to capture these shifts in reef-associated organisms in response to thermal stress. So not just looking at corals, but the entire reef community and seeing whether or not these communities are undergoing shifts throughout bleaching events and the recovery. So coral bleaching is a well-studied phenomenon where prolonged stress can actually lead to coral death. So in some cases, this may cause entire reef communities to shift towards algal-dominated systems. And there's actually a recent bleaching event in 2019 in the summer and late summer and fall of last year, which had its thermal maximum in around September. And that provided a unique experience to actually track using eDNA what's happening on the entire reef scale. Now, this is particularly important because many studies only look at how bleaching is affecting the coral animal themselves. So we know that coral bleaching is stressful to the corals themselves, but much less attention has been given to the cryptofauna. So all those little critters hiding inside the nooks and crannies and how they react to bleaching events. So these cryptic species actually make up the majority of reef diversity, and therefore bleaching is vital for conservation of reefs. So now for some of the specific methods we used in this project. So for my study, I wanted to balance somewhere in the middle of those methods of meta-barcoding and species-specific primers. So I actually developed primers that targeted only Hawaiian corals as a group. So corals that are in the Hawaiian Islands, and developed primers specifically for them. And I actually built 2 sets of primers just to kind of help with this replication. So we targeted ribosomal 16S and cytochrome oxidase 1 areas of the genome. So that will be referred to as 16S and CO1 respectively from here on out. And the location of these relatively short, so only 100 to 120 base pairs of DNA, very small fragments. You can see they're actually located here on the mitochondrial genome of Poeciloptera damicornis. So again, very short regions of the genome itself. And I decided to begin these eDNA surveys of coastal field sites around the island of Oahu. So I picked 4 sites representing a wide range of coastal development, and all that were extremely shallow and located just offshore in order to reduce costs. All these sites were accessible from shore, and I didn't need any boats or scuba equipment, teams of divers, nothing like that. So the first one was Mokualohe, the marine refuge in Kaneohe Bay, which is the highest % cover of coral that we found among our 4 sites. I also picked Lanikai and Waimanalo. These sites were adjacent to popular recreational beaches and are made up of patch reefs. And then finally, I picked Wailupe Reef in Maunalua Bay, which had the least amount of coral cover, and it was actually representing the site with high levels of urbanization and coastal degradation. So at each of these sites, I performed visual surveys, and these visual surveys were modeled after state monitoring agency protocols, so very similar to like what NOAA would do or DLNR out on the reefs. So at each site, % coral cover was quantified using 3 20-meter transects, and then half meter by half meter quadrats for photos, taking a photo every half meter or meter along the reef. And then these photos were blind allocated and analyzed in coral point count software, which overlays 100 random points per photo and basically just identifies what species under each point. And that's how we visually calculated % benthic cover on the reef. And then in order to compare eDNA to the visual surveys, we then collected seawater at each of those same sites. So 4 samples of seawater were collected from the site, and these were taken from 1 meter above the reef substrate at the same area as the visual surveys were conducted. And a primary concern with using eDNA is actually contamination. So it's very easy to contaminate DNA from your samples. So we monitored that at every step of the way by taking these negative controls into the field with us. So this is kind of what the sampling would look like. We collect some of the seawater in bottles like this, and we bring negative blanks into the field. So just no DNA in it, just filtered tap water basically to monitor for contamination. So samples were then transported back to the lab on ice where we filtered them, and all of our equipment was sterilized with 20% bleach to minimize contamination. We then extracted the DNA from the filters and amplified them using PCR, polymerase chain reaction of our 2 genes, using our negative controls as well throughout this process. And then the DNA was sequenced on a Illumina MiSeq platform. So this is all very nitty-gritty molecular ecology. I'd be happy to talk more about these methods in detail, but kind of glossing over this at this point. DNA sequences were then filtered, grouped, and matched to online databases. So basically taking all of these little barcodes of DNA sequences and matching them to published DNA sequences from coral genera that we know and recognize. So now for some of the results from this. After the reads were paired, merged, and quality filtered, the majority of assigned eDNA sequences were from anthozoans, as you can see here. So anthozoans are anemones, soft corals, hard corals. So basically, the primers that I designed to amplify corals did in fact amplify corals. So that was a good thing. There was only slight differences between our 2 separate primers, 16S and CO1, which are colored black and blue respectively here. But both primers together, actually, they amplified a total of 78% of stony corals. So most of the eDNA that we got back was from stony corals. And the remaining metazoans that were classified from our coral primers were actually predominantly assigned to sponges, which incidentally had a similarly conserved genome at these same locations. So they just happen to have very similar sequences to corals, so we also got some sponges. All right, so now that we know that our coral eDNA primers worked, can we actually use them to quantify coral cover out on the reef? So from our 2 survey techniques, we wanted to see if total eDNA, which you can see here on the x-axis, can predict percent cover from visual surveys, which would be here on the y-axis. So if this was actually possible, we would expect to see a linear relationship between the coral DNA reads and the coral percent cover from visual surveys. So using both 16S and CO1 from Kaneohe, just to start, we can see that this is in fact a log linear relationship. So the solid line in the middle represents the best fit relationship, and the dashed lines on the outsides represent the 95% prediction intervals here. The same goes for when we add our data from Lanikai, Manalua Bay here in green, and Waimanalo here in purple. So overall, using both genes together, the coral DNA reads explained 91% of the variation in coral cover from visual surveys. And this was a highly statistically significant relationship. So we can, in fact, get coral percent cover from eDNA. So now breaking it down by gene, looking at only the 16S genetic marker primers that we used, that actually explained even more variation with an R-squared value of 0.94. So that explains slightly more variation than using both genetic markers together. And therefore, CO1 actually explained the least amount of variation with an R-squared of 0.71, although this was still statistically significant. So these prediction intervals you might notice are much wider on average, meaning that the CO1 marker is actually slightly less accurate when determining percent cover of corals. So now we know that we can effectively get estimates of percent coral cover from eDNA from only a filtered bottle of seawater. But how about looking at the entire community of reef-associated organisms? So again, back to this picture of the reef here. We know what's happening in terms of corals, but using eDNA, can we detect any differences in bleaching events that they're having on the entire reef? So the entire reef community. To do this, we sampled 3 times throughout the 2019 bleaching event. So you can see here early bleaching in July, mid bleaching in September, which was actually the thermal maximum, and then late bleaching in November, as represented by these vertical green dashed lines. And here the horizontal red line actually represents the bleaching threshold for most corals. So above that point, most corals will start to bleach. So you can see that our early bleaching coincides with before anything would have bleached, mid bleaching pretty much at the thermal maximum, and late bleaching as that curve is starting to level off and go back down again. So the results from the eDNA community analysis are going to be plotted in what's called an NMDS plot, where these communities that are in this compressed 2-dimensional space. So again, communities that are more dissimilar to each other will be separate, so not really overlapping. As you can see here, community A is very different from community B. If these communities are more similar to each other, they will be overlapping in this 2-dimensional space. So again, community A is overlapping community B, so these are very similar communities. All right, so using our actual data, the NMDS plotting revealed that our 4 coastal field sites were significant, having very few overlaps in community structure. So Waimanalo and Lanikai were perhaps the most similar sites that we surveyed, whereas Mauna Lua and Kaneohe were perhaps the most distinct reef communities that we surveyed. And this is pretty obvious if you've ever been to Mauna Lua Bay or Kaneohe Bay. Kaneohe is dominated by a high coral cover, as well as plenty of fish, rays, and other organisms, whereas Mauna Lua Bay is pretty much dominated in a lot of areas by invasive macroalgae and may have few or less actual fish species, invertebrate species, things like that. In terms of the response to bleaching, communities during early bleaching were significantly different than those from mid-bleaching and late bleaching. So you can clearly see here that the communities narrowed, and these became significantly less similar during the thermal maximum, which was during the mid-bleaching. So again, everything's kind of shifting from the early bleaching, very diverse community, kind of getting more narrower towards the mid-bleaching, and then somewhat bouncing back towards late bleaching. So now that we know that reef communities are shifting throughout these bleaching events, let's take a look at which groups of species are actually being affected the most by bleaching. So here we have a bar chart of marine phyla across the x-axis, so all of those broad scale groupings of species, and the amount of eDNA from the filtered seawater will be represented on the y-axis. So the colors here show the grouping based on which stage of bleaching they were surveyed in. And so the green is pre-bleaching, red is mid-bleaching, and purple is post-bleaching. And these markings above the bar chart represent which groups had statistically significant differences at different stages of these bleachings. So I'm going to talk a little bit about specific groups here. So our first group of interest would likely be chordates. So this is organisms such as reef fish, mammals, rays, all of these things appear to increase in relative abundance during the height of bleaching. Corals, on the other hand, and other cnidarians such as, again, anemones or jellyfish, also have a similar trend with significantly more eDNA being produced during the height of the bleaching event. So this could actually be perhaps due to an increased shedding of coral mucus during this stressful time for corals, and they're actually contributing more eDNA to the water because they're stressed. Another group of interest would be mollusks. So this could be organisms, anything from cone snails, cowries, sea hares, bivalves. All of these organisms had significant reductions after bleaching began. So the eDNA signal actually dropped off substantially. And on the other hand, porphirids or sponges had a significant increase after the bleaching, perhaps suggesting that these sponges are having a positive response to coral bleaching, whereas most other things are having a negative response. All right, so let's try and make some sense of all of this. So in short, from simply a bottle of seawater, we're able to successfully demonstrate that meta-barcoding approach can actually provide reliable estimates of coral cover on Hawaiian reefs. There were also highly statistically significant relationships between visual estimates of percent cover and what we got from coral eDNA. So eDNA can capture differences in relative abundance among our field sites, which can be seen here in photoquadrats from each of our 4 sites: Lanikai in the top left, Kaneohe in the top right, Waimanalo in the bottom right, and Manalua in the bottom left. So all of our sites had varying degrees of coral cover, yet eDNA was able to capture these relative differences quite easily. And in terms of response to bleaching, we have evidence to support that coral reef-associated organisms do actually undergo community restructuring during bleaching. So that actually suggests that even sublethal bleaching effects, such as this most recent bleaching event in 2019, which didn't actually cause that much widespread death compared to previous bleaching events, but even these sublethal bleaching events can affect the entire reef community. More specifically, we found that there are fewer arthropods, vertebrates, mollusks, and corals after bleaching began. However, there appears to be an overall increase in sponges. That being said, some limitations are that we don't really know the effective volume of water that's being surveyed from eDNA. As you can see here from this drone shot that I took from Maunalua Bay, all these patch reefs, we don't actually know which patch reefs are contributing to the eDNA signal. So if I took a bottle of water from the center here, I would assume that it's coming from this area, but again, the exact area that we're surveying remains unknown when we take a bottle of water. Also, these transport dynamics are poorly quantified for most coastal sites, so we don't know whether or not wave action is affecting this. It probably is, but also currents and local hydrodynamics are playing an important role in how eDNA is affecting coral surveys. Also, the reproducibility over time is not well established, so this is among the first studies where we're actually looking at a long time series of data to see how the signals in eDNA are changing over time. And standardization of protocol is needed because most eDNA studies are using drastically different protocol. And unpublished data we have from using these small volume 60 ml syringes to collect water actually yielded drastically different estimates of coral cover when we compared that to using larger volumes of water. So that just goes to show you that just the standardization of the protocol is needed in order to obtain accurate results. We also know very little about how biological factors such as growth rates, excretion rates, reproduction rates, all of these things may be affecting eDNA signals over time, especially excretion in terms of coral bleaching. So as we saw, the coral mucus actually may be contributing more to the eDNA signal if you happen to be taking your water samples during coral bleaching events. You may get an overrepresentation of coral. There may also be seasonal effects of these rates. And this worked well in a relatively simple system. So here in Hawaii, we have very few like branching complex coral taxa. So it's unknown at this time whether or not this same method would work in areas where we have a higher abundance of branching corals, such as this image you can see here of a crop or a stand in the Caribbean. So much more surface area of coral. We don't know if this would actually throw off the relationship. As a survey tool though, regardless, we're able to rapidly assess the coral percent cover here on Hawaiian reefs. And these primers designed specifically for corals were able to differentiate the coral genera here in Hawaii. And actually they provided a more accurate estimate of diversity when compared to visual survey techniques. So this is rather unsurprising, as there are many studies that have looked at observer biases in visual-based survey methods. So you can imagine here in this picture, if all of these divers are writing down different survey results, whether or not they're unsure of a visual identification of corals or something like that, if each one of these divers is collecting a different visual data, then you can see how the results might be skewed a little bit. And this is actually a problem that occurs a lot in visual surveys, where every visual survey member is trained to the same degree. So eDNA surveying is highly economical, costing approximately 90% less than a single visual survey of a similar coastal survey one on Oahu. Whereas in contrast, it took me around $444 to survey one entire reef on my own, just from a bottle of seawater. So this actually is, can increase the accessibility so this can increase the
Your screen just went off.
Can increase the accessibility of monitoring efforts and actually
Your screen just froze, Patrick. Can you hear me?